Okay, and it's not on yet, of course. Okay. Okay, so let's go ahead and get started walking through the pre-final. So the internal ribosome entry site in viruses allows viruses to skip the initiation step of translation. They can direct recruit the ribosome. So which of these can now be ignored, right? Which can be absent and translations still occur for this? Yeah.
Translation initiation.
Yeah, so we can lose the initiation factors. It can still bind there and begin translation. Does it still need elongation factors? Yes. Yes, so elongation factors are still necessary, right? Because just because the ribosome can be recruited there and binds and begins translation, it still needs to do elongation at every step. It needs to add a new tRNA and add a new amino acid at every step. So it needs all the regular elongation machinery. Does it need a release factor? What does the release factor do? Based on the name, what do you think the release factor does? Yeah.
Spread it. Like to hold it like.
Yeah, it separates the ribosome, right? So the release factor binds to the stop codon and releases the ribosome into its two halves. That's still necessary here. The only thing that this lets you skip is normal initiation. So if we're skipping normal initiation, do you think we still need the initiator tRNA? What does the initiator tRNA do? What amino acid is bound to the initiator tRNA? Methionine. So it recognizes the start codon, right? The anticodon of the initiator tRNA recognizes the start codon. The viral genes still have start codons. So even though we're skipping ordinary initiation, we still need an initiator tRNA. We still need to do regular start codon read through, or regular start coding reading right there, right? So all of these are necessary except translation initiation factors. Yeah.
Why is translation initiation factor necessary?
So the initiation factors primarily serve to recognize the cap structure, the 5' cap structure, and pull in the ribosome to that location. So if we have an internal ribosome entry site, we're not recruiting to the cap. So we can skip cap binding and begin initiation just with a 40S, just with a small subunit at the IRES, instead of needing all of the initiation factors for cap binding and scanning. But we still need the initiator tRNA in order to recognize that start codon and translate the gene normally. If we didn't have the initiator tRNA, it wouldn't decode a methionine, it wouldn't do the actual large subunit joining effect, and it wouldn't translate the gene properly. Any questions on this one? Okay, so which strategies would block SARS-CoV-2 replication without harming the host cell? So first of all, which of these would harm the host cell? Yeah, so if we mess up ribosome assembly, the host cell is not going to do translation, the host cell is going to die. What else? Right, so DNA-dependent RNA polymerase, that's POL2, right? That's POL2 and the other RNA polymerases, POL1 and POL3, but mainly what we're talking about here is POL2, right? We're not going to be getting regular mRNA synthesis, we're not going to be getting regular gene expression. Both C and D would kill the host cell. So would inhibition of an RNA-dependent RNA polymerase affect SARS-CoV-2 replication? I see some nods. So why? Why does SARS-CoV-2 need a RNA-dependent RNA polymerase? Exactly, yeah. Its genome is RNA, right? So it needs an RNA-dependent RNA polymerase to both make more of its genome and make its mRNA, right? So SARS-CoV-2 is a plus-strand RNA virus, right? So it itself, its own genome is also the mRNA, but it makes a negative sense using an RNA-dependent RNA polymerase, and then uses that negative sense as a template to make a bunch of the positive sense, both as new genomes and as protein vectors. Okay, so what if we targeted the SARS-CoV-2 genome with siRNAs? What would happen to the genome? Degradation, yeah. So just like any other RNA in the cell, you target it with an siRNA, it gets associated with the RIS complex, you get cleavage, and you get destruction of that RNA. So both A and B are viable options for blocking SARS-CoV-2 replication without harming the host cell. Any questions on this? We don't do that. Like, we don't have any RNA-dependent RNA polymerases encoded in our genome because it's not a function we do. We never make more RNA off of our RNA. Okay, so mRNAs used in vaccines are designed to include which of the following to allow protein production inside cells. So what is different about mRNAs in vaccines compared to normal mRNAs in the cell? Let me put it a different way. Do you think the FDA is going to let me put stuff into human cells that is not similar to stuff that is already in human cells? These are essentially identical to real mRNAs. So they'll need a 5' cap structure for stability, a poly-A tail for stability, and an open reading frame encoding whatever protein you want to produce. Will the mRNA itself have introns? No, right? All the introns for an ordinary mRNA would have been spliced out long ago. For these, they never existed. We just put it in intron-free, it goes into the cytoplasm, it gets translated, makes the antigen, your body produces antibodies against that antigen, and then you will be immunized against that the next time your body sees that antigen. Any questions on this one? Okay. Which of the following happened during splicing of pre-mRNA in eukaryotic cells? So let's go through it in order. Does U1 snRNA base pair to the 3' splice site? I saw one head shake no. That's right. It doesn't. It base pairs to the 5' splice site. Does U2 snRNA base pair to the 3' splice site? I saw one no. It doesn't. Where does U2 snRNA base pair? At the branch point, right? So that's part C. U2 snRNA base pairs to the branch point, right? So C is correct, right? And then D. What's a transesterification reaction? So that's one of them. Yeah, so during that ligation, that's one of them. The transesterification reactions are when you have a phosphate that has an oxygen, right, attacking a different phosphate at the phosphoid point of another nucleotide, right? So then you result in a new ester being made. And the ester is phosphate, oxygen, phosphate, right? So that's the phosphodiester bond, right? And those phosphodiester bonds, those new ones that are made, need to be made twice during intron splicing. The first one is to the branch point. The second one is the ligation of the exons together. So for this one, it's C and D. You need U2 snRNA base pairing and 2 transesterification reactions. There's the one that goes to the branch point from the 5' split site, and it makes that intron loop, that lasso that she showed before. Okay. Which of the following increase the chances of DNA translocation events? So what's a translocation event? Yeah, so basically 2 different chromosomes swap pieces, or just a piece of a chromosome gets ligated to a different chromosome. Yes. So how does that happen? What has to happen for a translocation to occur? Yeah. Yes, 2 double-stranded breaks, right? So one spot needs to be broken, and another spot needs to be broken to be an acceptor for that piece. So, if a small guide RNA that you design is not specific and cleaves in multiple locations, does that allow for DNA translocation? Yes, right? So if you're cutting in multiple places in the genome, you get multiple double-stranded breaks. Any of those breaks serves as valid acceptors for all of those pieces to swap around. What about a drug inducing high expression of transposase? Yes, right? So every time a transposon moves, it's creating double-stranded breaks when it cuts that out. Those are supposed to get ligated back together and fixed, right? They don't always. And if that's happening at a very high rate at multiple different places in the cell, because we have a lot of transposons, there's a high likelihood of 2 of those broken spaces being translocated during the transposon moving. Okay. Would nucleotide excision repair cause a double-stranded break? No. It just cuts out the nucleotides that are the problem, and only on one strand. So you don't have a double-stranded break, you don't have the possibility of translocation. What about mismatch repair? How does mismatch repair work? So it changes a couple nucleotides, right? So mismatch repair recognizes when the newly synthesized strand of DNA is problematic, and it removes a large chunk of that newly synthesized strand and then fills that back in. But during all that, right, it's just removing one strand. We don't have a double-stranded break. So this also would not increase the chances of DNA translocation because we're only removing one strand. Yeah?
So for A, what would it be if it's a small interfering RNA, like where it matches up and then it can eat away at the location? Would that be different?
So does siRNA target the genome? Right. So the RNAs are going to be targeted there. The RNAs are going to get degraded. There's going to be no breaking of DNA in that case.
So like with the siRNA though, it just leaves the RNA piece right there, right?
Yeah, it only attacks RNA, which is out in the cytoplasm. It doesn't even get into the nucleus. Okay. So how does RAS work? What does RAS do? Yeah.
It's a GTPase. So when bound to GTP, when activated by like a gap, then it's in its activated form, and then causes a phosphorylation cascade.
Yeah, so RAS is a signaling protein that causes a phosphorylation cascade that leads to cell growth, right? And in the case where it's overactive, aberrant cell growth, right? Tumorigenesis. RAS is active when bound to GTP, and then it hydrolyzes GTP to GDP to inactivate itself. So it itself is a GTPase that turns itself off. And this is how the cell regulates so that you don't have overactive RAS expression and tumorigenesis. So what would happen if we reduce the GTPase activity? Yeah.
It would, is it E, we're taking away the activating portion of it?
Yes. Yeah. So B is also correct, right? If we reduce the binding to GTP's activating protein, so GAP, we don't get as much GTP activity. RAS stays GTP bound, right? And it's active, so we're going to get more tumorigenesis. That's the same as having reduced GTPase activity. So A and B are functionally the same, right? So those are both correct here. If we had reduced binding to GTP, would RAS be active? No. If we have increased GTPase activity, would RAS be active? Yes, but briefly, right? So RAS is still going to go active, right? It's going to act as a GTPase faster. It's going to spend a lot less time in the active phase. We get lower RAS activity, lower cell division. Yeah.
Sorry, so the ACE part, it's generically like an activator, right? So like ACE means it's an activating protein, or?
So ACE means that it acts upon that to hydrolyze it, right? So a DNase hydrolyzes DNA, cleaves DNA. An RNase hydrolyzes RNA, cleaves RNA. A GTPase hydrolyzes GTP, breaks it down into GDP. And when RAS does that, it turns itself off. Okay. Which of the following would you need for a CRISPR experiment designed to change a glycine to valine in the active site of RAS in lung culture cells? Yeah.
Homologous DNA template.
Yep, so you need a template, right? So you're going to provide a template, so you're going to swap the valine codon in instead of the glycine codon. How does that template get swapped in? What's the first thing that needs to happen for that to be a viable option for the cell? Yeah. Yeah, you need to actually cut that DNA, right? So you need the Cas9, and then how are we going to target Cas9 to the right place? With the guide, right? So A, B, and C are all necessary, right? Would an siRNA do anything to help us here? No, right? It's just going to degrade the RNA, doesn't touch the genome at all. Any questions on that? Yeah. A, B, and C.
Did you say D was yes or like no, and it's just for short time?
Right, no. It's A and B. Yeah.
The small gut RNA, that is very interesting, but how do we know or like, do we understand that part of the small gut RNA is that part where exactly Cas9 is going to go because it's basically near there, Cas9 is going to.
Yeah, so any cut that is near the region of your homologous template is going to be a viable option for replacement, right? Because the first step for homologous repair is that the ends get chewed back by 3' endonucleases anyway, right? So it's going to have these overhangs that can then invade and synthesize new from the homologous template. So it doesn't matter, it really doesn't matter where your cut is at all, as long as it's close. Like you can go about 500 base pairs in either direction and still get pretty highly efficient recombination. The closer to where you want, the better it's going to be. Okay. So how are we going to know that we actually made the edit in the cells that we're interested in, right? If we use Sanger sequencing to sequence the RAS gene in the edited region and compared to the database, would that tell us if we've made the edit? Right? So we use Sanger sequencing, we directly look at that, right? Similarly, if we use next-gen sequencing, we would get the whole genome. This would tell us both whether we made the edit and whether we had substantial off-target effects, right? And then could we use RNA sequencing to look at the mRNA population? If we've changed the codon in the genome, is the mRNA sequence going to be different? Yes, right? So RNA sequencing would tell us the same thing. So all three of these sequencing methods would tell us whether we successfully made the edit. Would PCR for the target site, followed by cell electrophoresis, tell us whether the edit was there? No, why not? Yeah.
PCR just tell us like there's high.
So the PCR just tells us the presence of the gene. Right? So it doesn't tell us anything about binding. It just tells us if that region of the genome is there, right? And it tells us the size, right? So it tells us the size of the region of the genome we amplify. If I swap one codon for another, have I changed the size? No, right? So it's a 3 nucleotide deletion followed by a 3 nucleotide insertion, basically, right? Nothing has changed. So it's A, B, and C. Any questions on this one? Okay. This next one. Okay. This one you'd have to pull out a codon chart. I have one open, but I don't think it's that useful to like jump to it. So a single nucleotide mutation in the active site of RAS that causes valine to be encoded instead of glycine is common, right? Using the genetic code, which of the following codons could be converted from glycine to valine by a single nucleotide change? So the first question I would ask myself looking at this is, which of these actually code for glycine, right? And I would go to my genetic code, and I would look at it, and I would see that all 4 of these are glycine codons. So that's a check, right? All 4 of them are currently glycine, right? Now, which one of them could be made to be valine by changing one nucleic acid, right? A single nucleotide change. All of them, right? So if you look at your genetic code, maybe I will actually pull it up. just hard to get in and out of this slide. Where's my mouse? There you are. Can I get you on my screen? Yeah, it's in presenter view, so it's giving me trouble. There it is. Okay. So, all of the GG ones code for glycine, right? All of the GU ones code for valine, right? So I could change any one of these by just changing the central G to a U to go from glycine to valine. So all 4 of them are valid answers for this one. Yeah.
On the test, we're gonna have like a amino acid chart.
Yes. Yeah. She'll give you a chart on the, I think on the front page of the final.
Also,
Mm-hmm.
two questions like on this one, like related. In the pre-final, there were like 20 questions about.
Yeah, but they don't rely on each other, right? So like, this tells you all the information you need to know. Which of the following analysis would allow you to identify cells with the codon for glycine edited by CRISPR to valine, right? All the information is there. Which of the following reagents would you need for doing a CRISPR experiment to do that same edit? All the information is here, right? So you don't need these two, but yes, generally her tests are themed around one gene and doing similar things to that one gene. Any instance where you have like data that you would need to answer both questions, that data will be repeated on both questions so that regardless of the order, you have the information you need. Okay. When you sequence the RAS gene from a benign tumor, you discover a single nucleotide change, G to A, in exon 2, which encodes the active site for GTPase activity. Which of the following could explain how this mutation results in less active RAS? So this question mainly focuses on how this may act as an exon splice regulator, right? So if this mutation disrupts an exon splice enhancer, would that result in skipping or retention of that exon? What does the exon splice enhancer do? So if you have an exon splice enhancer, it enforces retention, right? The exon splice enhancer tells the splicing proteins the boundaries of this is an exon, recognize this, right? So if you mutate that exon splice enhancer, you would get skipping of that exon. Yeah.
So is it kind of like opposite? Because like when I see like enhancer, I think like okay, like oh, it would be like opposite of an intron, right? Like take this out, I think.
Hold on. Can you repeat that?
So like if it said like intron splice enhancer, that would mean like it would want to take like that part out and just really actively try to, right?
There are no intron splice enhancers.
No, I know, but like if you were to consider like oppositely, does that make sense?
There's no proteins or binding sites that function as intron splice enhancers, right? There's only exon splice enhancers, and the exon splice enhancers tell the cell this is a proper exon, right?
So leave it alone.
So retain it, right? So it tells it this is an exon, tells it these are the intron boundaries at the sides of this exon. Okay, yeah. Okay, so if the mutation creates an exon splice silencer, you would get skipping of the sequences that encode the RAS active site, right? Hold on a second. Yeah, so A and B are correct, where if you have a mutation disrupting a splice enhancer, you will get skipping of that exon. If you have a mutation creating a exon splice silencer, you would get skipping of that exon. And then for C is the opposite of A, right? The mutation creates an exon splice enhancer, which would not result in skipping of the sequence, right? And then D, the mutation changes the codon for lysine to aspartic acid. This is where you need to go back to your codon chart and check, and both of these are still lysine codons. So that's not true. So this one is just going to be A and B. Yeah. What do you mean by do they always, right? So like an exon splice silencer promotes the skipping of the exon, right? But it's sort of like everything in the cell is based on levels and things kind of fighting with each other, right? So a single exon might have multiple exon splice enhancer regions and exon splice silencer regions, and then it depends on what proteins are available in the cell to actually bind to those regions and tell it whether to listen to the silencers or to listen to the enhancers. So both are typically present in most exons, and that allows for different cell types to do alternative splicing. Yeah.
So is the silencer just inhibiting like the splicing proteins from binding to the exons?
Is the silencer? It can be. It's a little indirect. There's generally other proteins that recognize the silencer, and then those proteins inhibit the splicing machinery from recognizing the nearby introns. Yeah.
Why is this not correct?
Why D is not correct? Because these both encode lysine. If you check the codon chart, they both encode lysine, so there is no change from lysine to aspartic acid. Yeah.
Was the underway of the nucleotide there, is that just to like highlight that they're different or like?
Yes.
Yeah, okay.
Yeah, just to say that's the one that is mutated. Any more questions on this one? Did you chase that down at all? It seems like just an error on the key, right?
It seems like it's just an error on the thing. Like you said, it's like the opposite of A.
Yeah, there's no way. I feel like I'm going crazy. All right. To identify spliced isoforms of RAS in the human lung tumor tissue, which of the following reagents would you need to synthesize cDNA off of the RAS mRNA? Yeah.
A, B, and C.
Yes. So you need a primer, right? And you always need a primer for DNA synthesis. You need the reverse transcriptase itself. That's going to use the RNA as a template, extend that primer to make the first single strand, right? And the only way it can do that is by having free DNTPs, right? Free deoxynucleotides to add to the extending nucleotide chain. You do not need NTPs, right? Because those are not incorporated into DNA, right? They are not deoxynucleotides. They are just nucleotides. Yeah.
Did we ever like contemplate NTPs, like the RNA or
RNA.
RNA?
Yeah. RNA is NTPs. Yeah. It's just not deoxy. It's just ribose. Okay. So, if you used RNA-Seq to compare transcriptomes of normal and lung tumor tissue, which of the following could you learn? So, would you get the number of transcripts with higher expression in the tumor compared to normal lung tissue? I don't even think it's a connection issue. That's the weird thing. It's like it still knows when my laptop is plugged in because it reacts to it. Oh, it's up there. It's not on my screen anymore. So if you did this RNA-Seq, would you get the number of transcripts with higher expression in the tumor compared to normal tissue? I saw some yeses and some nos. So yes, you would get this, right? So you get total reads of all the RNA in the cell. You can look at all of the transcripts, right, and see which ones are upregulated in the lung cell, right, and just count those. And you can do that all programmatically. Okay. Would you get differences in alternative splicing in the tumor compared to normal lung tissue? Yes, right? So you would see all those sequences. You can look at which intron or which exons are skipped, which introns are potentially retained. You would get all of those reads, and you can map that back. Would you see differences in alternative polyadenylation in tumor compared to normal lung tissue? What would it look like if you were looking at the genome browser and a gene had different polyadenylation? Yeah.
It would be like some kind of mutation, right? Because it should always have, at least if it's the same cell type, it should have the same amount.
So you might see a mutation, right? So the polyadenylation signals, some of them are retained in the 3' UTR. So you might actually detect the sequence difference there. You can also see that it's missing, for example, the last exon, or it's missing a portion of the last exon, right? Can the last exon be skipped by alternative splicing? No, right? So the only explanation for that would be alternative polyadenylation. So you would also get that information from this. And then would you get the number of proteins with higher expression in the tumor compared to normal lung tissue? No, right? This doesn't actually tell you anything about protein abundance. You can infer that it is more likely for the protein to be more abundant if there's more mRNA of it. That is not always true. They correlate better than like correlating DNA to protein level, but they don't correlate perfectly. Any questions on this one? Okay. What happens if there's an intron in the 3' UTR of a gene? What gets left behind when that's spliced out? Right. So there's going to be an EJC dropped into the 3' UTR. So A is true, right? And then what is the consequences of an EJC being downstream of the ribosome stop site? Deadenylation and decay, right? So that's going to be recognized as a problem, right? The cell treats that as a premature stop codon. It's the way the cell has designed to look for mRNAs with premature stop codons is, hey, another exon was ligated in downstream of this, right? Like that's a problem. So that's looked for and that's destroyed, right? Would that result in lower levels of the RAS protein? Yes, right? So the RNA is unstable, it's getting degraded. And then finally, would the RAS RNA produce higher levels of the RAS protein? No, right? There's no mechanism in place for the RAS RNA to suddenly boost that expression. It's just getting destroyed, there's just less of it. So this is A, B, and C. Yeah.
Can we also, so like the translocation, that's like where we have like the promoter and like specific transcription factors like that are important there too, right?
So no. Transcription factors are almost always going to be upstream in the 5' region, and they'll be completely upstream of the 5' UTR as well.
Okay, so like the 3' UTR, oh sorry, I think it was like backwards. Yeah, okay, yeah.
Any other questions? Okay. So you want to look at possible mutations in the RAS gene from 2 lung cancer patients. You do PCR using primers from a region in the gene from tumors and nearby healthy tissue from each patient. What kinds of changes in the RAS gene in the tumor cells could make it oncogenic? So In order for this to be oncogenic, right, we know RAS promotes cell proliferation, right? So you get, if you're a tumor, and you want to get more cell proliferation, you're going to want to turn RAS on in some way. So is it possible that the RAS gene is being amplified in the tumor of patient 1? Yes, right? We get a darker band on the PCR that indicates that this patient's tumor has duplicated the RAS gene at least once in the genome. So there's a full extra copy of the RAS gene, most likely. And then for B, right? What's going on in patient 2? We see in the healthy cells that this element that we're amplifying is 800 base pairs. So what happened in patient 2's tumor? Just based on size. Yeah. There's a deletion of something, right? So again, for the tumor, right, overactive RAS is what we're looking for. So would overactive RAS in the tumor be explained by deletion of a negative regulatory element? Yes, right? So you delete a negative regulatory element, now you get more RAS. So, conversely, right, looking now down at D, does deletion of a positive regulatory element in the promoter make sense for this? No, right? If you deleted a positive regulatory element, you would have less RAS, less cell proliferation. Now looking at C, integration of a transposon into the RAS gene in the tumor of patient 1. Does that make sense with the data you've been given? No, right? There's no increase in size, right? If there was a transposon that was inserted there, you would see an increase in size in this region, and you just don't see that. You just see an amplification of the region, more abundance of the region. Yeah?
So if it's said for C, like in patient 2, would that be right then? Because there's 2 different genes showing up there?
No, because the healthy region is bigger. Like the healthy normal one, right? That region is bigger than what we see. So it's only explained by a deletion, not by an insertion.
Okay, so like for both of them, like the tumor at 8 and then the tumor at 6?
So if you look at the healthy cells of the same patient, right? You amplify this region, you get an 800 base pair piece, right? In the tumor, you amplify that region, you get the 800 base pair piece, meaning 1 copy of RAS is normal. You also get a 600 base pair piece, meaning 1 copy of RAS has a 200 base pair deletion.
Okay, so you add most likely. So you use like
There's no insertion there, right? Nothing has increased in size from the healthy. Yeah.
And how did you know it was negative, a positive regulatory element?
Because RAS needs to be turned on more to be oncogenic, right? So you have a deletion, right? If you deleted a negative regulatory element, that would give you more expression of RAS. So that's the only deletion that is consistent with this data. Yeah.
So the band is just indicating expression, but not size.
So the band is indicating number of copies of RAS in the genome, right? Because since you're amplifying DNA, you're not looking at expression at all. You're not seeing RNA levels. You're only seeing DNA amounts. So if you started with 2 copies of RAS, which is what a normal cell should have, right? You get a band that is that dark, right? And then if you do, if you take tumor 1, right? You get a darker band. That means you started with more DNA in the PCR. That's why she has this note at the bottom that says, note that the same concentration of genomic DNA was used for each PCR. That means for the same amount of genomic DNA in the healthy cells, from the same amount of DNA from the tumor cells, you have more copies of the RAS gene. Yeah.
So back to like tumor 2 for patient 2, you have 600 and 800. So you said like we've lost 800, we lost 200 in the 600. Is that what you're saying?
Mm-hmm. Yeah, we lost 200 base pairs from RAS.
Okay, but would it be like adding because the patient 1 doesn't have that 600?
So patient 2 in the healthy cells, right, has an 800 base pair band. So the mutation in the tumor cells is due to...
The addition of a 600.
It's not addition, right? The 600 base pair band is showing up because one of the 2 copies of RAS in that cell has lost 200 base pairs.
Otherwise, it would just go back up to 800.
If it was duplicated. Okay. Constitutive activation of RAS leads to changes in alternative splicing of several different genes. You look at northern blotting to analyze splicing of gene L in lung cells expressing 2 different RAS mutants. Based on the below gene structure of gene L, which receives a 200 nucleotide poly-A tail, and the northern blot results using a probe for exon 3, which of the following conclusions are consistent with the data for RAS? Whew. Okay, so you have this gene, right? If everything is appropriately spliced, you will have exon 1 is 100 base pairs, exon 2 is 100 base pairs, exon 3 is 100 base pairs, exon 4 is 150 base pairs, so that's 450 base pairs with 200 poly-A, 200 nucleic acids added as a poly-A tail. So that gives you a 650 base pair mRNA, which is exactly what we see in the wild type cells. And then if you look at the next one, right, you see that you have much less of the RNA and that it's bigger, right? So yeah, we're specifically talking about mutant 1 here, right? So what explains the RNA being bigger in size? Would intron 2 being retained explain that? Yes, right? So intron 2 is 50 base pairs. We've moved up from 650 to 700, right? Now, why is there less of it, right? She's highlighted that there's a stop codon in intron 2. The mRNA is subjected to nonsense-mediated decay because of this premature stop codon, right? So just like I talked about previously, the ribosome terminates there. There will now be an exon junction complex downstream of that termination that will not be getting cleared, and that will result in nonsense-mediated decay. Okay. Is exon 3 being skipped? No, right? We see it still on the blot, and because our probe binds to exon 3, that means exon 3 is still there. Can the mRNA be unstable because it is fully spliced but not polyadenylated? Is that in line with this? What size would it be if it was fully spliced and not polyadenylated? 450, right? It's not 450, it's up at 700. So C and D are inconsistent with the data. A and B are consistent with the data. Any questions on that? Yeah.
So the 550, what if it looks like the poly-A was like, I don't know, halfway, like 100 times, or that wouldn't matter because it would be able to get out of the nucleus?
I mean, if the poly-A was 100 nucleotides instead of 200?
Yeah, because we have like 550, so like instead of like 650, would DE right then, or no?
No, because because the mRNA is still larger than the wild type baseline. If the mRNA was unstable because it was fully spliced but not polyadenylated, it would be shorter than the wild type baseline, regardless of the length of the poly-A tail.
So then, like, what is the 550 band on RAS2? What is that?
We'll get there. That's a different question.
Oh.
It's been correct in your point, and then the activated using the AGC.
Using what?
And it's starting to conflict.
Yes. Yes. So there's a premature stop, right? There will be an exon junction complex downstream of that. This mRNA will be destroyed. That is nonsense-mediated decay. Okay, which of the following conclusions are consistent with the data for RAS mutant 2, right? Okay, so now looking at this one, we have RAS mutant 2, we have the 550 base pair band, or nucleotide band. Would this be consistent with exon 2 being skipped about half the time? Yeah.
I had a question about the last.
Sure.
Why is there still
It's unstable, but it's not made, right? So like it's made, it's unstable. There's less of it, which is why the band is thin, because it's getting degraded rapidly, but it's not being degraded instantly. So whenever I freeze the cells, I collect the amount that was stable at that time that hadn't been degraded yet. Okay, so if I skipped exon 2, what's the size of the RNA now? 550, right? So exon 2 being skipped about half the time, why does she say about half of the time? Yeah. You see two bands and they're about equal in intensity, right? So about half of the time, exon 2 is being skipped. That's in line with this data. What if it said exon 3 is skipped about half of the time? Would that be consistent with this data? No, why not?
Because then you wouldn't see any probe.
Yes. So if exon 3 was being skipped, because our probe is for exon 3, we wouldn't see the lower band, right? We wouldn't be able to detect it because it's missing exon 3. Okay. Yeah, so since exon 2 is being skipped about half the time, B is also correct by definition, right? Alternative splicing is resulting in 2 different mRNA isoforms in these cells, right? That's literally what we're detecting here. Exon 1 is skipped about half the time. Exon 1 is also 100 base pairs. Is that consistent with this data? Why not? I saw you shaking your head, so now you have to answer.
It's because I'm pretty sure for alternative splicing, we can't splice out the first one.
Exactly. You can't skip exon 1, you can't skip exon 4. So is D correct? No, right? Exon 1 is skipped half of the time, and exon 4 is skipped the other half of the time. That also doesn't make any sense, right? You can't skip exon 1, you can't skip the last exon. Any questions on this one? Yeah.
So it's A and B.
Yes.
And then, so first off, hold on, can you see the splice and this nucleotide kind of hanging on to the first part of the mRNA, and then
No.
how does it?
No. So the whole mRNA is going to be made, right? It's going to be exon 1, exon 2, intron 2, exon 3, exon 4, 200 nucleotide poly-A tail. That's what we're seeing in mutant 1, right? That's what's being made. The ribosome sees the stop codon when it goes to translate this mRNA and terminates right there when it's halfway up the mRNA, right? And the cell recognizes that as a bad thing. It recognizes that as premature termination and targets that mRNA for destruction.
Okay, so the ribosome just like stops, and then it just keeps doing its own thing, and then that's when the cell comes back and introduces it into replication, right?
Yes. Okay. The 12th codon in RAS is often mutated in lung tumors. Using the genetic code and the below sequence, figure out what the amino acid of the 12th codon is. Basically, you're going to scan through this till you find the AUG and then start counting, which I can't do on a slide. I can annotate. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, so you'll just count out codons, get to the 12th codon, go to your genetic code, and translate that that is glycine. I remember that from a few minutes ago when all of the GGs resulted in glycine. But yeah, so it's a pretty straightforward one, but it's a little weird to do in a review. Any questions on this one? Yes. Yes, the start codon is the first codon. My laptop's dying. Really can't even hold a charge for 3 hours, huh? Pardon you? Okay. All right. Switch from okay. You're studying tumors from 2 different patients and find that they both contain the same point mutation in RAS that causes it to be constitutively active. This is what we would expect, right? Overactive RAS means a tumor, basically. To learn if both tumor types have matching gene expression profiles, you perform DNA microarray experiment, comparing expression from each tumor to normal lung tissue from the same patient. Again, remember when reading these, because they're a little confusing to read, especially in grayscale, right? Look at your key, right? The not detected here is black, right? So that means it's not present in either. The tumor is white, so that means it's present much more in the tumor or not at all in the normal, right? The same in tumor and normal would mean that that spot is yellow functionally, right? So that's the dotted one. And then the striped one is normal, so that would be going the other way, right? So if you labeled tumor green and you labeled normal red, the striped ones would be red, the white ones would be green here, right? That's the way I always have to think about these to read these in grayscale, is I think about, like, I go through and I literally, if I was doing this, I would write tumor green, normal red, that one, same in tumor normal, yellow, and not detected black, right? And I would just write that every time, because I see actual microarrays, I know what they look like, and it's just hard to translate into grayscale. Anyway, so you're looking at expression of the genes present in tumor, present in normal, same in both tumor and normal, and not detected overall. Okay, so what would be the expression pattern that you would expect for an oncogene? Yeah.
A tumor to show up?
So what would be the expression pattern, though, on the microarray?
I think you said green, right, for tumors?
Yes, so it would be present in the tumor, right? And exclusively in the tumor, or much more abundant in the tumor, right? So those are oncogenes. They are active more in tumors. So based on these results, which of the following genes are good candidates for acting as oncogenes in patient 1? Yeah. Sorry. You're good. Go ahead. Yes. So A, C, and D are all upregulated, specifically in the tumor in patient 1. They are potential candidates for oncogenes in this. B is not. It is more present in the normal. So because it is the same in the tumor and normal, you wouldn't consider it a good candidate for an oncogene, right? That's being expressed, say there's 100 mRNA molecules in normal cells and also 100 in tumor cells. Is that likely to be an oncogene? Right, it's no different. Yeah.
If we ask for proto-oncogene, then would that be?
No. So like the problem is you can't know a thing is a proto-oncogene until you've seen it become an oncogene at least once.
Okay, so you can't like infer.
No. You have to know something about the gene's function, not just know its expression levels to determine it's a proto-oncogene. Okay. Moving on to patient 2, right? So, yeah, these are, I just realized that they're like, they could be A, B, C, D in order, but anyway. Which of the following genes are potential candidates for acting as tumor suppressors in patient 2? So what's the expression pattern we would expect to see for a tumor suppressor? More abundant in the normal, right? It's something that's being lost in the tumor. It's ordinarily supposed to be suppressing the tumor. You've now lost it. So you would expect it to be more abundant in the normal. So which of these have decreased expression in the tumor and therefore more expression in the normal in patient 2? B and C, which is A and B. But anyway, B and C, right? So weird thing happening here, right? Seems like C can be a potential oncogene in patient 1 and a potential tumor suppressor in patient 2. This probably means that this gene is not a good general candidate for drugging, right? Because it behaves differently in different individuals. But yes, B and C, which is answers A and B, is correct here. Any questions on this one? Okay, same setup. Based on the below results, which genes should be targeted by a small molecule inhibitor as a cancer therapeutic for both patients? So what type of genes would I want to inhibit in a tumor cell? Yeah. Proto-oncogenes or oncogenes, right? So the actual tumor-causative ones, I definitely don't want to inhibit tumor suppressors, right? So I'm looking for an oncogene that is present or seems present in both patients. So I'm going to target A, right? So I'm going to try to make a small molecule inhibitor for gene A. I wouldn't target the other oncogenes, like I said about C, right? C and D, right? They may work for patient 1, they won't work for patient 2, because they're not being overexpressed in patient 2. Yeah.
And as you said, there have to be more in-depth to separate out what's going on.
Yeah, F just looks like a normal gene, right? F just looks like something that the cell kind of always needs, right? And the cancer cells don't upregulate it compared to the normal. It could be something like actin, like just the cytoskeleton of the cell, right? The cancer's making the same amount of actin as a normal cell is because both cells just need actin.
So it's not going to work out.
You don't know anything about it. You only know its expression level is the same in both cells, right? Which means it's not a good candidate for looking further into for why is the tumor different from regular cells. Because really, when you're doing these experiments, that's always what you're looking for is what's different about the tumor compared to normal cells. Yeah.
So, what's the difference between
So proto-oncogene is a gene that can lead to cancer when mutated, right? But it's the regular gene that's in your cells right now, right? Like it's the regular version of it. Like RAS in everyone's cells right now, for the most part, unless anyone has cancer, is a proto-oncogene, right? If RAS gets mutated, now you would call it an oncogene. If it gets mutated in a way that actually causes cancer. Until you know some information about it, right? And I know a lot of information about RAS because a lot of people have done a lot of studies about RAS. Yeah. Almost never, right? So like you would almost never chase that down as being anything interesting, right? It's gonna be something, like I said, something basic. Or it's gonna be like actin. Yeah. So if it's present in the tumor and in the normal, it's definitely not doing anything that is cancer specific. Okay. You want to sequence this commonly mutated region of the RAS gene. What are the first 4 nucleotides of the PCR primers you would use to amplify this entire sequence? So if you want to amplify this entire sequence, reminder, the 3' ends of your primers need to point into the region that you are trying to amplify, right? This is a double-stranded DNA. It's going to peel apart. You're going to want 5' ACCC, right? The beginning of this top strand, because that primer will now bind to the bottom strand. The polymerase will extend the 3' end to make this whole strand. The converse is true for the reverse, right? When that peels apart, you need a primer that will base pair 5' on the far right, 3' towards this way, that will then get extended and make this whole region. So you need the complementary region to the 3' end of this, which would be CACA. Any questions on making those primers? Yeah.
So, for these, I would, in my head, say, okay, like, we're making a complementary strand, right? So then, like, on the 5' end, it would be GGG, and then inside it would be CACA, and then split them apart, and then, so it's basically the same thing.
So TGGG, right? Because DNA strands need to base pair anti-parallel, that would be pointed the wrong way. It would get extended going this way. So like if I have my DNA, right? And this is what she's given you in the problem. It's just the top strand. There's also a bottom strand, right? So when you do the denaturing, your primers are going to anneal. If you provide it primers that are complementary to right here, this primer is going to look like this, 5', 3'. And the polymerase is going to make that, which is not the region you want to amplify. The polymerase is going to bind here and extend that way. So you're not going to make the DNA you actually want. You need primers that bind here, 5' to 3', and then extend this way, and here, 5' to 3', and then extend this way. So you need a primer that's complementary to the top strand at the 3' end, and a primer that is identical to the top strand at the 5' end.
And is that always for like any primer, no matter what we're doing?
Yes.
Okay, so then, so like 5' is basically the same, 3' is complementary.
Yes. So like the region you want to amplify, if this is what it looks like, you make one that is identical to the 5' end and complementary to the 3' end.
For any kind of primer.
Yes. Okay. You're good. Yes. Okay. This next one, I do also want to write out. Okay. All right. Activated RAS leads to upregulation of BCL2 for the sequence of the BCL2 gene, for this sequence, where the +20 and the -20 sites are underlined and bold. Which of the following reagents would be used to make a probe for the detection of BCL2 mRNA expression levels in a northern blot? Okay, so the first thing we need to do with this one, which is a little bit complicated, is, where did I put that eraser? Is figure out which strand we're even looking at, right? So, you have your 5' end, and that's labeled +20. It's T, right? And then G, T, G. This is awful. T, G,
It's
T.
T, G, T, G, T, G, T, G, 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 C, G, A, G, G, E, G, T, G, 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 A, G, A, A, G, G, A, G, G, A, G, G, T, and then again T, and then I got the reagents.
Thank you. Yay. Okay. Cool. So, am I looking at the template or the non-template strand? So, this is +20. Let's just say +1 is somewhere in here. I don't want to count it right now, right? So, that means the RNA is going to be made in this direction. RNA is going to be made 5' to 3', but this is going 3' to 5'. So this must not be the strand that's the same sequence as the RNA, right? So therefore, it's the template. It is the template that will make the RNA. The RNA will start, let's see, 20, 19. So close. So the RNA is going to start being made right here, right? It's going to be made 5' to 3'. T, C, T, C, C, T, C, G, C, C, A, C, C, A, C, A, C, A, C, A, 3'. Right? So now we're looking at it, right? This is the +1 of the RNA. This is the +20. Yeah. Yes. It's only 3 of them. Thank you. But yeah, so that's going to be synthesized there, right? So this is the template strand. That is the RNA sequence. Okay, so of these, right, easily, the easiest one to answer yes to here, right, is B. We're going to need a radioactive label for the probe, right? So we're going to use a probe to detect this mRNA. So the next question is, which of these sequences is actually going to be complementary to this RNA? Which of these sequences is going to be a valid probe that's going to bind to this RNA and light up on our northern blot? Right, so the original region of the template strand that is on the plus side is going to be complementary to this. Even though this region on the template strand is also listed up there, right? It's not a valid probe for the RNA because the RNA doesn't have this sequence. The RNA starts here, right? So it's A and B. Go ahead.
So you said template strand is not the same sequence as
Correct. So the template strand is the reverse complement because it's used as the template to make the RNA. Yeah.
How do you know it was the templates?
Because of the directionality, right? So the plus 20 here is up at the 5' end, right? I count back to the plus 1. I can go ahead and label that, right? And then I know the RNA needs to be made 5' to 3', starting at the plus 1 and going to the plus 20, right? So if I had been given the reverse of this, right? Or this is the plus 20, this is the minus 20, then this wouldn't work, right? I would need RNA that is made, why am I using my hand? Need RNA that is made going in this direction, but this direction is already 5' to 3'. So I would need RNA that's being made 5' to 3' off the opposite strand, which would now be identical to this. So you need to think through with those pluses and minuses, which direction is 5' to 3', and match it to the direction the RNA needs to be synthesized in. You have a question?
Yeah, so since this strand is the reverse complement of what
Mm-hmm.
what you want to be made, you just use
Yes, you just use the beginning of this sequence, right? You can't use C, right? So C is also the same as this sequence. It's just on the other side because it's in the minus region. It's not going to bind to the RNA because the RNA is over here, going from plus 1 to plus 20. Yeah.
So you said if we go backwards, with the plus one over here and minus over here, why is it complementary? And why is it identical to the complementary for the other side?
So if I switch it, right, if I just draw this in cartoons without sequence, right, and I have a strand that's 5' here, 3' here, plus 1 here, plus 20 here, right? I need RNA that's made going from plus 1 to plus 20 in a 5' to 3' direction, right? So my other strand is going to be 5' over here, 3' over there. This is going to peel apart, and now the polymerase needs to pick one of these strands as the template and synthesize in a 5' to 3' direction. So in order to make this, the polymerase needs to bind here and synthesize this, which is going to be identical to the top strand. Everything that is synthesized in the cell, nucleic acid-wise, is synthesized 5' to 3' directionally, right? So in order to synthesize 5' to 3', because two strands of nucleic acid can only be anti-parallel, they have to point 5' to 3' in one way and 5' to 3' the other way in order to bind, right? This is the only way you could synthesize a 5' to 3' piece when this strand is given in this directionality. Yeah.
So A is correct because we're keeping it to that 5' direction.
So A is correct. A is correct because it will bind the RNA that is made starting here, right? So if this is +1 and this is +20, right? It's already written 5' to 3', right? So in order for the RNA to be made 5' to 3' going this way, it's as easy as the polymerase binding here and synthesizing in this direction. So it's going to make a sequence that's complementary to this, and you want your probe to be complementary to the RNA, right? So since that's the region that is being copied into the reverse complement, that is also the region you will use as a probe. The complementary strand is always there, right? It's always a DNA duplex. In reality, for convention, we write it as a single strand because it's easier to work with that in like computer programs and everything. Truthfully, in reality, if I was like doing anything real with this, I would just use the other strand and write it so that the plus direction is going 5' to 3' already. Yeah. Oligo is the Roman variant of poly. So it just means multiple. Greek versus Roman. Poly Greek, oligo Roman, but it just means oligonucleotide, so many nucleotides. You order these, we call them oligos because they are many nucleotides. Yes. Yeah, I know that's a pretty complex one that you kind of have to dig through a lot of the reverses. Any other questions on that? Good. Tom's yelling at me. Okay, so now we're looking at the RAS 3' UTR. It's predicted to contain LET7 microRNA target sites. To identify these sites, you do a series of reporter genes containing the 3' UTR with regions mutated. You compare the expression of the reporters in culture cells plus and without an inhibitor of LET7. So, which of the following are consistent with these results? So let's just walk through what we're looking at first, right? So clearly, if you look at the full 3' UTR, right, you put that in a reporter cell, you add the inhibitor of LET7, and you get more expression. So immediately we know, right, versus plus inhibitor versus minus inhibitor, immediately we know that LET7 is regulating this 3' UTR in some way, right? Because when you add the inhibitor, you get more expression, meaning that the LET7 microRNA is ordinarily suppressing translation off of this 3' UTR. So how do we narrow down the regions, right? So start with A. There's a LET7 target site within position 250 to 300 of the RAS 3' UTR. That's going to be whatever's happening in that overlap region that's in 2, but not in 1 and not, or sorry, yeah, that's in 2 and not in 1, right? So in that little overlap region with that mutagenesis, suddenly the expression is the same both with and without inhibitor, right? So we've broken the regulation by LET7 by mutating that region. And it's similar for 3, right, which you would expect because 3 also has that same region mutated. So A is correct here. There's a LET7 target site within position 250 to 300. Any questions on how I arrived at that? Yeah.
Can you explain how you know that there is the LET7 target?
Yeah. So when that region is mutated, right, when I've mutated 2, the expression without the inhibitor of LET7 goes up, which means ordinarily LET7 is doing something at that position, presumably binding, right? binding and causing downregulation, going from 5 to 2, right? But when I've mutated that region, it is no longer downregulating that expression. And I know that it's in that region specifically because when I mutate 1, which also mutates all of the other regions in 2, I don't get that expression change. So I know it's specifically between 250 and 300. Yeah.
So A is correct.
Yes.
But like in the practice, in the brief final, it was false. It was marked as wrong.
Yeah, I mean, I don't have.
Only G type.
That's the first answer. They're in a different order. Okay. Yes. So let7 target site. So yeah. So be careful when looking at the pre-final. Apparently, it reorders the letters. Okay. Sequences from 500 to 550 contain a negative regulatory region that is regulated by something other than let7. So what are we looking at to look at 500 to 550? We're looking at effects that happen in 5 that don't happen in 4, right? That's going to be this little overlap right here, right? And you see that expression both with and without inhibitor goes up by 2, right? So the inhibitor becomes irrelevant for the change that made. So this is also true, right? There's something in there that is usually suppressing expression, and then we've deleted it, or we've mutated it, and we get more expression, and it doesn't depend on let7, because whether I add the inhibitor or not, I get an equivalent increase in expression. First.
So with let7, there's less of the cells down here.
There's less of the cells. So this is just expression, right? So we're looking at how bright the cells are, because this would be attached to like GFP. So we go from the regular reporter, right? If we look up at the top, it has an expression level of 5 with the inhibitor, 5 with the inhibitor and 2 without the inhibitor, right? I mutate this region, and now I have an expression of 7 with the inhibitor, right? So something changed that is not in a let7 dependent way. Even though let7 is inhibited already, I still get an increase in expression when I mutate that region. And I see the same increase in expression from 2 to 4, so plus 2, when I don't have the inhibitor, which means whatever is negatively regulating, whatever is negatively regulating expression is not dependent on let7 in that region. You were next.
So whenever you add expression, so like we went from 5 to 7, we're basically adding 2, it's always going to be negative.
The regulator that you've just deleted is negative. Yeah.
I have 2 questions. So the first one is, we're going to be looking, like from the beginning, you want us to look like down the 2 columns with the numbers, right? And that's how we can kind of see like what's going on as we increase the mutations, right?
Down the 2 columns.
So like the black bar is the mutation. So if we're looking like 5, 5, 5, 5, 5, 7, we can see like automatically something happen in that 5 to 550, right?
Yes.
And then if we look to the negative, so it's not like 5, 2, 5, 2, 5, 5. It's like row by column, right?
Yes. I mean, you should take the whole data into account, right? So like clearly if you look at 2, right, 2 and 3, right, they don't have an expression change with the inhibitor, but they do have an expression change without the inhibitor. So the effect that the mutations in 2 and 3 are having are dependent on let7.
Okay, so like if you see like 2 and 3, because they match with and without, we can automatically say like the mutation there isn't, mutation does the same like expression, right? Because there's nothing different between them.
Well, the expression level this is just a quantification of the expression level, right? Like this has simplified it down where like you take a pool of cells, you've done this thing to them, and you look at how bright they are, and it's quantifying how bright they are, right?
Okay.
So it's saying when you see a 5 here, it means those are both a level 5 amount of GFP.
So it's not necessarily expression.
It is expression. It's reading out expression using GFP fluorescence.
Okay. So negative regulatory means it's not down-regulating, and positive regulatory means yes, it is regulating down.
A negative regulatory region is suppressing translation in this case, because 3' UTR. A negative regulatory region is suppressing translation. So when you delete it, you get more expression.
So negative regulatory is suppressing, positive is.
Yes, the function of the negative regulatory region is to suppress translation. So think of it this way, right? This is going to make an RNA. I prime, blah, 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 blah. It's going to have this UTR here, right? Ordinarily, in the 550 region, there's a something here, and a protein binds to that and says, whoa, don't translate, right? Something recognizes that region and says, don't make more protein, right? And then what I'm doing in 5 is I've deleted this. So now this protein can't bind, and now there's no inhibition of expression, and I get more protein made, right? So whenever you delete something and you get more expression, it used to be a negative regulator, right? You lost the negative regulator. So yes, so if I delete something and I get more expression, it was a negative regulatory element.
So it's like negative, like no, it's not regulating anymore?
So it's a negative regulatory element because its purpose and its ordinary purpose is to suppress expression. And when I delete it, it no longer is there to be suppressing expression. Yeah.
So if we went from, let's say, 5 to 3 to 1,
If we went to 5 to 3. Yes. Right? So if this dropped from 5 to 2 to be like 3 and 0, right, you would say that that was a positive regulatory element. Because I deleted it and I lost expression. Yeah.
Why is it something different than what?
Because the inhibitor doesn't have any effect. Right? So like the inhibitor of LET7, if you look at 2 and 3, right? In the absence of inhibitor, when you delete that region, expression goes up from 2 to 5. With the inhibitor, expression doesn't change, right? So the inhibitor is here to tell us whether the effect is LET7 dependent. Without the inhibitor, LET7 is active. LET7 is able to do stuff. And yet, the expression of those goes up, which means you've deleted a LET7 region. But because it's specific to the inhibitor, or specifically when you don't have the inhibitor, you know it's LET7 dependent in 2 and 3. In this one, whether you have the inhibitor or not, expression still goes up, which means LET7 is irrelevant for that question, right? So like LET7 is not regulating in this region because even when I inhibit LET7, I still get the increase in expression. And without the inhibition of LET7, I get the same increase in expression. Okay, and then C, there's a LET7 target site within position 500 to 550. This is not the case, again, because it is agnostic to inhibitor, right? It does not care whether the inhibitor is added or not present. It goes up by 2 in both cases, right? Sequences 500 to 550 contain a positive regulatory region that is regulated by something other than LET7. This is also not the case, right? It's a negative regulatory region. When I delete it, I get more expression. It's not a positive. So this one is just A and B. Yeah.
So target site means that something is done? That would be something's done?
So these are microRNAs, right? So a target site means somewhere where the microRNA binds and leads to destabilization or less expression. Any questions on this one? So this is basically the cloning step of how you would design this actual experiment, right? In order to design this actual experiment, you need to put it into a GFP making vector, right? So to identify which regulatory elements in the RAS3' UTR you need to design a reporter, which strategies would allow you to amplify the 3' UTR and correctly insert it into this vector for a GFP reporter. So this one is also a little confusing, and it helps to draw it out. So we're looking to insert the RAS3' UTR into this GFP vector. Okay, so we need, that's fine. So we have the RAS gene, 5', 3'. How do I want to do this? Yeah. Let's just call this the 3' UTR. Okay. Okay. So we want to insert it into here, and we probably want to directionally clone it, right? We want it directly after the GFP, and we want it to be in the same orientation, right? So we need to add restriction enzyme sites to this, and we're going to cut our vector with the same restriction enzymes that we add to this 3' UTR. So in order to insert this, where are we going to put a 3' UTR in this to see how it affects translation? Yeah.
After the GFP?
Right after the GFP, right? So at that multiple cloning site where EchoR1, BAMH1, and HIN3 are, right? That's where we're going to put it. Okay? So we need 2 different primers that are going to amplify this region and in a particular direction, right? So we'll need either EchoR1, BAMH1, or HIN3, and we'll need them in that order. So whatever we put on the left, if we put EchoR1 or BAMH1 on the left, we can do EchoR1 and BAMH1, we can do EchoR1 and HIN3, and we can do BAMH1 and HIN3. We just need to make sure that the one we're putting on the left is upstream in the vector as well, right? So if we want to put BAMH1 on the left, then we have to put HIN3 on the right. If we put EchoR1 on the left, we have our choice of BAMH1 and HIN3. We just need to make sure it's the same order. Like the order they're in there has to be the same order we put on this fragment. So to amplify this, we're going to need a reverse primer that binds here, right? So it's going to have a region that's complementary to the end of the 3' UTR, and then has our restriction site of interest on the 5' end of the primer, right? So when primers go to amplify, you can add whatever you want to the 5' end, because it's the 3' end that needs to actually bind to whatever DNA you're amplifying. So you can add restriction sites up here, but you need this. And then in order to amplify this, right, we're going to amplify it from DNA. So this is the reverse complement of the 3' UTR. We're going to need a primer that binds here and has our other restriction site. Okay, so that's what we're going to make. So let's see if we can generate something usable from these answers. Use a PCR primer with the EchoR1 sequence at the 5' end, followed by a sequence complementary to nucleotides immediately after the RAS stop codon. So by definition, right, the UTR comes after the stop codon. So the stop codon is going to be right here. So if we use the 5', so if we use 5' echo R1, right, and then sequences complementary to nucleotides immediately after the RAS stop codon, right? So it's complementary to nucleotides immediately after the RAS stop codon. Is that going to bind the complementary strand? if it's complementary to this, will it bind here? No, it needs to be identical to this. So these nucleotides would not work to amplify this. It's a very confusing question. This one might also have issues. Hold on, give me a moment. Okay. Her key says yes on that, so I'm gonna work through this. I don't think that's the case, to be honest. Are you seeing what I'm seeing, Tessa? We're doing it live. We get to work through the question together. I know. I'm looking at the key. I just don't agree. Okay, so use a PCR primer with the echo R1 sequence at the 5' end, followed by sequence complementary to nucleotides immediately after the RAS stop codon. That doesn't work. It needs to be identical to the sequence, because it needs to be identical to this in order to be complementary to this, to actually make that strand. Yeah.
What if it's at 3' end? Then would it be right?
You never want to put a restriction enzyme site at the 3' end.
Because we need it to bind.
We need it to hybridize, yep.
Okay.
So we can immediately eliminate C and D. So yeah, so the key has A and B. I think that's just an error given how confusing this question is, to be honest. I'll talk with Dr. Pasquinelli about it and check on it, but I'm pretty sure they would need to be identical to this region of the 3' UTR, right? They would need to be identical so that they base pair to the reverse complement and synthesize that region. And then, yeah.
Are we going to have something on Monday? Like,
Probably. I'm taking the final on Monday, so I'll find out.
but like I understand like the vector part, but like so basically like just like like we can do whatever to 5' end, 3' has to null, and then it has to be complementary to the 5.
Yes, you can do whatever you want to the 5' end as long as your 3' end of the primer base pairs to the region you want to amplify. Okay. Use a PCR primer with the HIN3 sequence at the 5' end, followed by sequence complementary to nucleotides downstream of the polyadenylation signals. This one is correct. This would be this primer right here, right? So we would have HIN3 site added right there. And then this region right here would be complementary to regions downstream of the poly A signal, because the poly A signal is part of the 3' UTR. So that would amplify that up. And then to make this perfect, I would change the wording on A to say, use a PCR primer with echo R1 sequence at the 5' end, followed by sequence complementary to the other strand immediately after the stop codon, right? So we would need sequence complementary to sequences immediately after the stop codon, or I guess the best way to say it is just sequence identical to the beginning of the 3' UTR. So sequence identical to nucleotides right after the stop codon. So yeah, I'll talk with her about that and make sure that's okay for the pre-final. Maybe get that question tossed.
So A and B or just
I mean, the answers she wants are A and B, and I see what she's going for, right? Like if you just think about it as like, yes, I need sequence that will base pair in this region, like yes, but it needs to base pair to the opposite strand. So So it should be identical to the nucleotides following the stop codon. You guys are welcome to correct me if you don't see where I'm going.
So the way I'm reading it is because you're enhancing like the DNA with inserting the vector, it doesn't. I mean, you need it to have like the right directionality. Maybe the complementary is like a little bit weird, but the idea is like you're trying to use PCR to generate the 3' UTR of RAS.
Yeah.
And then stick it in with the EcoR1. So it's a primer that will have EcoR1 at the 5' end, and then the 3' of the 3' ETR. Yeah, so I feel like it's a little bit weird, like identifying directionality and like having it. You would need 2 primers to have.
She's trying to say with this that you need a primer that has echo R1 at the beginning and then binds to this region, right? And she's just saying it very generally because she's thinking of the DNA as being double-stranded. And she's like, oh yeah, it'll be complementary to one of those 2 strands.
If you combine the 2 answers together, then you would have
Yes.
EcoR1 and N1, and that would be your 2 planking sequences.
Which is correct. Like that is the order you would want to put these in. You would want eco R1 and then N1 or N3 downstream.
Yeah, I think just not having the sequence throws me off because like my brain wants to like go find the sequence
Right.
and make sure they're playing each other right. Having it in just words, I feel like
This is hard to write out in just words. I get that
yeah,
for sure.
I think that's where I get confused.
Anyway, so big confusing question. The gist of it is you need to add these restriction enzyme sites to either end of the thing that you're amplifying, right? And you do that by putting Hen3 with sequences that are at this end, the 3' end of the UTR, and you put echo R1 with sequences that are at the 5' end of the UTR, which would be immediately downstream of the stop codon. That's what she's going for there. I take issue with the complementarity here, right? Because it needs to be the same as the top strand and therefore complementary to the reverse strand. So I'll talk with her about that. I will probably get free points for everyone on this particular question on the pre-final. Yeah.
Can you also see too, because BAM H1 is in the middle of the order GFP, that you automatically say, okay, it can't be C, an ND, it can't be D?
So you can immediately say it can't be C and D because it's trying to add the restriction sites to the 3' ends of the primers.
Okay,
But you could use BAM H1 if you wanted to. Like for A and B, let's ignore the complementarity, right? For A and B, echo R1 in A and Hen3 in B is correct. So is echo R1 and BAM H. So is BAM H1 and Hen3.
I thought you couldn't have a mutation enzyme if it like cuts the vector in half.
You need to cut the vector in half. All of those will cut the vector in half.
No, but what she's saying is if we had BAM1, we remove the GFP sequence.
Yeah.
Oh, I didn't even see that cut was there. Yes, don't use BAM H1.
Yeah.
Okay, so like that's another reason why we
I was literally only looking at the multiple clone sites. Don't use BAM H1 because that would cut between the promoter and between GFP.
So that's like a good way to say like, okay, like if it cuts it, then it can't be in there restriction enzyme, so D is out for sure.
Yes. D is out for 2 reasons. Yep. Sorry about that. Whew. Okay. Any follow-up questions on any of that? I know that was a lot. Okay. You put a GFP reporter with the RAS 3' UTR into normal and tumor lung cells and measure GFP expression levels. Based on these results, which of the following can be concluded, right? So we're taking what we made previously. It has the 3' UTR of RAS. We're putting it into tumor cells versus normal cells and looking at the expression, right? So compared to normal cells, tumor A has lost a negative regulatory activity that acts on the 3' UTR. This is true, right? So we suddenly get a lot more expression in tumor A, and the only thing that we're putting in here that's driving GFP is the RAS 3' UTR, right? So that 3' UTR is there. That's what's controlling the expression level of GFP here. And then in tumor A, we get much more expression off of this. This means there's usually some negative regulatory protein that is now not present in tumor A. Okay. In B, compared to normal cells, tumor C has gained negative regulatory activity that acts on the 3' UTR. This is also true, right? So that's suppressing expression of GFP, right? So it has something new there, new protein or more of a particular protein that acts in a negative way on the expression of GFP. Okay. Compared to normal cells, tumor A has gained a negative regulatory activity that acts on the 3' UTR. This is not the case, right? We have more expression in tumor A. If it gained a negative regulatory activity, it would look like C. And then similarly, D is the reverse of B, right? Tumor C has lost a negative regulatory activity. This is also not true because tumor C would then express that more. Yeah.
If C lost a negative regulatory activity that acts on the RAS,
Yeah.
wouldn't that make RAS more active?
Yes, that's D, which is wrong. B says gained. The pre-final puts them in a different order. Any questions on this one? Yes. Okay. Which of the following are possible explanations for the different GFP expression levels in normal versus tumor A cells? Where did that go? Okay. A microRNA that targets the RAS 3' UTR is not expressed in tumor A cells. Yes, right? So that's exactly like what the previous question was talking about, right? That's some negative regulatory activity that is now lost in tumor A cells. AU-rich RNA binding proteins that bind sequences in the RAS 3' UTR are degraded in tumor A cells. What do the AU-rich mRNA binding proteins do?
Deadenylation?
Yes. They lead to degradation and deadenylation. They lead to deadenylation and then degradation, right? So these would ordinarily be negative regulatory elements, right? And now they themselves are being degraded. So would you get more expression of RAS? Yes, right? So A and B here are correct. Repressors that target the RAS promoter are inhibited in tumor A cells. Does GFP here read out the RAS promoter at all? No, right? The only thing we put in was the 3' UTR, right? So we can't say anything about the RAS promoter. The RAS 3' UTR has positive regulatory sequences in tumor A cells and negative regulatory sequences in the normal cells. Have we changed the sequence of the 3' UTR? Not in this experiment, right? So in this experiment, we just put the GFP upstream of the regular 3' UTR. So the sequences are not any different. So C and D are incorrect. Yeah.
Can you explain what does that mean by degraded? Does that mean that it's gonna not have any
So the AU-rich RNA binding proteins are the proteins that recognize and degrade AU-rich regions of RNA, right? So those proteins would lead to degradation of the mRNA and less expression. Those proteins themselves are being degraded in the tumor cell. So when those proteins are lost, you now will get more expression, more stability of that mRNA.
And that's why we have less tumor.
No, that's why we have more GFP expression. It's one possible explanation for how we have more GFP expression, is that these RNA binding proteins that target AU-rich RNA for degradation have been lost in tumor A. So a negative regulator has been lost in tumor A, and we get more GFP expression. Okay. Which of the following steps would you use to identify if there's a homologue of the human RAS protein in C. elegans? A and B, right? So you're going to use BLAST to find if the RAS protein is similar to anything in C. elegans, and you're going to consider BLAST results with a very low p-value, right? So less than 10 to the minus third, not results with greater than 10 to the minus third, which is C. Yeah, so if you did D, right, you would need to copy the whole C. elegans genome and paste it into that little search box in BLAST, and then search that and see if human RAS comes up, right? That's not going to work, right? You're going to be scanning through millions of sequences that are the same between C. elegans and humans. You want to take the RAS protein from humans and use that as the query to blast against the C. elegans genome. Any questions on this one? Yeah.
Is the less than 10 is that like a generic thing?
Yeah. It's the standard p-value cutoff we use for blast searches. Yep. Okay. Which of the following would be needed for a successful RNAi experiment designed to inhibit an oncogenic RAS expression in lung tumor cells? Yeah. So we need Argonaut, right? So Argonaut is the protein that binds to microRNAs and targets it to the appropriate location. It's part of that RISC complex. And B, right? So we need the actual siRNA that will target the RAS mRNA. We don't need Cas9 endonuclease. We are not using a guide RNA to target the genome. And we don't need, sorry, we do need Drosha.
I think we need Drosha only the microRNA.
Yeah, you're right. Yeah, sorry. We don't need Drosha because there's no processing, right? We're not introducing the primary. We're introducing siRNAs that are already pre-cleaved. Drosha would be needed for the regular endogenous pathway, not for us to do an RNAi experiment. Yeah.
And I think that's only one factor, right? Like
Of the genome, right? So like Cas9 endonuclease is going to cleave wherever you target in the genome.
DNA, right?
Mm-hmm. Yeah. Okay. If ADAR edited a base in the RAS stop codon, which of the following would happen? What does ADAR do? Yeah. A, right? So the adenosine would be converted to inosine, right? Now you need to know what inosine looks like for base pairing, right? So inosine essentially looks like a guanine, right? It's going to follow all the same base pairing rules as a G, right? So now you go to your codon chart and you look for UGG, which is a which is a tryptophan codon, right? So B is also correct. Okay, the uracil is not going to be converted to inosine because ADAR acts on adenosines. And the change is not silent because it doesn't switch to another stop codon. It switches to a tryptophan codon. Yeah. So inosine is read as a G by the translation machinery. So you need to go to your codon chart and look for UGG. And that will encode a tryptophan. Okay. Which of the following would happen if you treated tumor cells expressing mutant-activated RAS with an inhibitor of the TOR kinase? First of all, I'm going to just give you A, right? Overall translation activity would definitely decrease. That's like the thing that TOR does. It massively upregulates translation activity, right? And then what is the consequence of that for tumor growth? It's going to be slower, right? So that's going to slow down tumor growth, right? And then C, would the rate of tumor cell division increase? No, right? The tumor is growing slower. It's not going to be dividing more often. And then D is the opposite of A, so that's also incorrect. So this is A and B. Any questions on this one? Okay. So for these, right, transcription of BCL2 is upregulated in tumors with activating RAS mutations. To study this regulation of BCL2 by proteins X and Y, you do DNase footprinting using this labeled region. Reminder, for those of you who saw me in discussion do this, the easiest way I find to do this is just flip that thing. So your label is at the bottom, draw it up like this, do minus 100, minus 80, minus 60, minus 40, minus 20, right? Flip that this way, and then look at your gel like that, right? So you have something like this, and now you can perfectly match those regions to where they would correspond in your reporter. Okay. So protein X binds to position minus 80 to minus 60 in the BCL2 promoter region. Yes or no?
Yeah.
Yes, right? So that's the regions that's being protected from the DNAs in this assay, right? They are not being cut here, which means you no longer get the fragments that look like this, right? You no longer get the fragments of this size range. Okay? The protein Y binding site is closer to the start of transcription than the binding site for protein X. Yes, right? The start of transcription is at the +1, so protein Y is more near the +1. Protein X and Y can bind the promoter at the same time. Doesn't look like it, right? You mix them together and actually they both lose binding. Protein X binds to position minus 20 to minus 40 in the promoter region. No, it's asking about protein X again. Protein X binds there. If it said protein Y, that would be correct. Yeah.
So the space is showing that it's binding into braided, right? Or not?
It's showing that it's binding, which means the DNAs can no longer cut there, and you don't get fragments of that size.
So the protein is there, right?
The protein is blocking DNA's cutting. Yeah. Okay. Based on the results of this IMSA, using radio-labeled BCL2 promoter DNA as the probe, and TBP, protein X, and protein Y, which of the following can be concluded, right? So what we're looking at with these IMSAs, right, we get labeled promoter DNA, we run it through a gel that under conditions that allows for protein binding, right? You mix that with different proteins, different isolates of proteins, and look at how much of that labeled DNA upshifts, right? So does protein X bind directly to the promoter DNA? No, right? So protein X is not resulting in a shift by itself, so it's not a direct binder. Does protein Y bind directly to the promoter DNA? Yes, right? So that's getting upshifted. Protein X inhibits binding of TBP to the BCL2 promoter DNA. Yes, right? So TBP will bind by itself. When you add protein X and TBP together, you no longer get binding to the promoter. Okay, and then D, less BCL2 promoter DNA is bound by TBP alone than in the presence of protein Y. Yes, right? So TBP alone, right? Some of the DNA is still here at the bottom of the gel. So some of it is unbound. TBP plus protein Y, all of the DNA is bound, right? So it captures more of that DNA. Yeah.
So B and C?
B, C, and D.
Okay, and then so when it's down, it doesn't matter like the size of the band, so like TBP, like you said, it means that some of it is being left behind, so it's not binding all by itself?
Yeah, so the size of it does matter. So the size of it there is meant to indicate intensity, right? So like this means all of the DNA is at the bottom, right? This means about half of it is still at the bottom, so about half of it is unbound. She just uses the thickness of bands to like indicate intensity in a black and white way.
So does it move fast necessarily, or it just means that it goes with it, not necessarily like speed?
The speed through the gel is this direction, right? So the lower it is on the gel, the faster it moved through the gel. Yeah. Yes. So TBP by itself, you get some binding to the promoter. You get some upshift. When you mix TBP and protein X with the DNA, suddenly TBP can't bind anymore. So something protein X is doing is inhibiting the binding of TBP to the BCL2 promoter. If this lane looked the same as this lane, then this would be false. So something about protein X probably binds to TBP directly and prevents it from binding to the DNA. Yeah. Why are there two bands? Oh, why are there two bands in that lane? So any DNA that TBP fails to bind will just run to the bottom, right? So this means that TBP can bind that DNA, but it's not a very strong binder. Like protein Y captures all of the DNA, TBP captures about half of it. Okay. Any more questions on this one? Yeah.
So running up the middle band of TBP is just straight TBP, and then the bottom one is the promoter DNA?
No, the top one is TBP and the promoter DNA. The only thing you see on here is promoter DNA, because promoter DNA is what's radio labeled.
Okay, so it's not like there's too much TBP to move on by itself.
No. TBP by itself, the protein by itself would be off the bottom of the gel, because proteins move through gels much faster than DNA will. Yeah.
Is also correct because there is more in protein Y compared to TBP.
So D is correct because of this lane. So in the very end lane, right, when protein Y is present, all of the promoter DNA gets bound by both protein Y and TBP, right? That's why it's higher up. It's heavier because it has protein Y and TBP in it, and you get no leftover DNA. So this could also be false if there were now two bands for TBP plus protein Y. If there was a very high band, that would be TBP plus protein Y, right? And then more DNA at the bottom.
So the bottom of the
Yeah, the promoter by itself is going to run to the bottom. That's why in the no protein case, that's where the band is. Okay. Okay. BCL2 is an anti-apoptotic factor that prevents cell death. To analyze the general chromatin structure of the BCL2 gene in normal lung tissue and 3 different lung tumors, you perform a DNase I protection assay using the southern blot to probe for a part of the BCL2 gene. Based on these results, which tumors are candidates for avoiding cell death by expressing BCL2? So basically, which tumors plausibly have expression of BCL2? Yeah. So tumor 1, right? So if we look at tumor 1, right, when you add DNase, you lose the signal for the BCL2. Let me double check that we're checking. It just says part of the gene. Okay, I was checking if we were looking at the promoter or just the gene. Yeah, so we lose the protection on part of the gene, right? So that indicates that it is in a euchromatic state. It is open. It is available to be transcribed, right? Tumor 3 is also a good candidate for avoiding death by expressing BCL2. Just because some of it is partially protected, it's still more open than normal cells. In normal cells, it's completely protected from DNase I. In tumor 3, there's a partial degradation by DNase I, which indicates it's partially euchromatic. Yeah. Because this is just asking about which tumors are candidates, you only look in the no drug condition. Yeah. We're basically ignoring the drug condition for this question. It's asking which tumors are likely expressing BCL2 to avoid cell death. Yeah.
So, when it's talking about avoiding cell death, so
So the tumor wants it to be expressed, right? So this is an anti-apoptotic protein, right? And when it's expressed, it keeps the cell from basically killing itself by many different mechanisms that recognize mutations, DNA damage, cancerous things, right? BCL2 can be overexpressed and then protect the cell from being destroyed.
Okay, so no band was expressed either.
No band means it was in euchromatin, right? So it's in an open chromatin state. It's not in the type heterochromatin. It has to be in an open chromatin state in order to be expressed, but it is not 100% guaranteed that it will be expressed. It's just asking, based on this data, which are good candidates, right? So it's in euchromatin, it's more likely to be expressed. If it's in heterochromatin, it's less likely to be expressed.
It's an anti-apoptotic factor, that's why. Okay. Right? That's why we want it to be expressed.
Well, I don't want it to be expressed. The tumor wants it to be expressed, right? So the tumor uses it to stay alive. Okay. Now, based on these results, where do we talk about the drug? Okay, which of the following are consistent? And then all of these ask about the drug, right? So the drug results in repressive chromatin formation at the BCL2 gene in tumor 1. Yeah.
Yeah, that's true.
Yep. Right. So we go from it being degradable by DNase 1 to no longer being degradable by DNase 1 in tumor 1, right? The drug does not affect the chromatin structure at the BCL2 gene in normal lung tissue. Right, so that's also consistent with this data, right? We see it's fully protected from DNase 1 in the control. It's the same level of protection in the drug case. How would this not be true? What would we need to see? We need to see it actually go up. Or go down, I guess. It says does not affect, right? So we would see it get degraded in the normal cells, or we would see an even darker, thicker band under DNase protection in the drug case. But yeah, so A and B are correct here, and then C, the drug results in open chromatin formation at the BCL2 gene in tumor 1. No, we already talked about the reverse of this. The drug results in increased expression of BCL2 in tumor 1. Again, no, because it's going into heterochromatin, right? So we would suspect that this drug results in lower expression of BCL2 in tumor 1. Yeah, so when you add the drug, right, we get more DNase protection, which means BCL2 is more heterochromatic, it's more condensed, it's less accessible to the transcription machinery, and we expect it to be less expressed. Yeah.
So in short, when you see the DNA stuff, band equals protection to heterochromatin, and then open is expressed, so the machinery can get to it, which means it's
Yes. Okay. Okay. So same expression or same DNase protection assay. In the controlled no drug conditions, you would detect more reads for the BCL2 gene from the normal compared to tumor 3 samples when treated. If you perform next generation sequencing in the DNase protection assay, right, would you expect more reads for the BCL2 gene from the normal compared to the tumor 3 samples treated with DNase? So if you did the DNase protection assay, right, you're destroying that region if it's in euchromatin. So it's no longer going to show up by next-gen sequencing either, right? So in the control samples, you would detect more reads for the gene in the normal compared to tumor 3, because tumor 3 has partial DNase degradation. Any confusion on that one? Yeah, so in the control no drug conditions, right, so left side, when you treat with DNase in tumor 3, you get degradation of the BCL2 gene, right? If you do next-gen sequencing on this sample instead of doing a northern blot, right, you would get less reads in the tumor, right? So you would get more reads in the normal because it's being destroyed by DNAs in the tumor sample.
So when we do next gen, we're not using DNA.
In this instance, you are. If you perform next-gen sequencing in your DNase protection assay, right? So if you do the DNase treatment and then do next-gen sequencing, you would get more reads in the normal and less reads in the tumor. Okay. Okay. Would the sequencing reads for BCL2 include promoter sequences? Yes, right? It's part of the DNA, right? We're doing DNase protection and sending it off for sequencing. The promoter region is going to be more likely to be more open, more likely to be degraded, but clearly this gene is not fully open in the normal cells. So we would get the reads from the promoter regions when sending it off for sequencing. Would the sequencing reads for BCL2 include exons but not introns? No, right? It's going to still have the introns, right? We're sending off DNA for sequencing. Okay? This is not like cDNA synthesis. This is direct chromatin DNA. In the control no drug conditions, you would detect less reads for the BCL2 gene from the normal compared to tumor 3. That's the reverse of A, so that's not true. So this is A and B. Yeah.
So for A, it's another reason why, because it's partially open, right? There's also been degraded, but tumor 3, so it's like half open, does that make sense, right?
Yeah. It's like partially open. A lot of it is getting degraded. So you would get more reads in the normal cells than in the tumor cells. Yeah. Yes, when you lose a band in this assay, you would consider that open chromatin. In this assay. You would expect to see the reverse in like a northern blot, right? So the northern blot, you would expect to see much darker bands in the tumors, or in tumor 1 and tumor 3, because that's more open. It should have more RNA expression. Okay, so we're looking at some ChIP-PCR, which is not a thing that people do very much, to study how 3 different drugs affect this regulation. You do the ChIP assay to analyze the association of specific proteins with the BCL2 promoter. Yes. Okay, so based on these results, which drugs repress the expression of BCL2 in the presence of progesterone, right? So we can just sort of look at this assay, right? The things we're looking at right here is the amount of DNA we get out, or just the presence in this case, because there's no variance in the thickness of the bands. So just, do we get the promoter out when we pull down using antibodies against these particular proteins, right? So which drugs repress the expression of BCL2 in the presence of progesterone? What's the easiest way to look at this and assay expression? Yeah. You can look at methylation, right? There's something that's even more direct. TBP binding, right? So if TBP is bound, you're getting transcription, right? So expression is active in the presence of progesterone under no drug conditions. It is active with or without progesterone under drug 2, and it is inactive with drug 1 and drug 3. How come what? That's true of all of them. without progesterone in the normal cells, you also have, it's also methylated. Yeah, so without progesterone in the normal cells is how it's supposed to be, right? Like no drug treatment, right? Without progesterone, it's methylated, it's inactive. With progesterone, right, it is now bound to the progesterone receptor, it is bound to acetylated H3, and it is bound to TDP, and it is no longer methylated. Yeah, so I would just immediately look at TBP as a proxy for expression in this assay, if she gives you TBP. If she doesn't give you TBP, then you need to look at acetylation and methylation. So methylated H3K9 would indicate that it is condensed, heterochromatic, not expressed. Acetylated H3K9 would indicate that it's open, right, and available for transcription. Yeah.
So it's drug 1 and 3 because it's testing it, so we don't have the papa binding. But if it was saying like which one, like if it was opposite, like which drug regular don't repress, then it would be no drug in 2, right? Because we have papa binding and they're
Yes.
open.
And in fact, drug 2 makes it active all the time, even without progesterone, which is kind of weird. Yeah.
For this, the progesterone receptor is not present. So, because the question didn't ask, does the progesterone need to bind this receptor? That makes drug 3 right.
So for this question, it's just asking, is there expression, right? Clearly, in drug 3, you don't get histone 3 acetylation, and you don't get teta-binding protein bound, which means there's no expression.
Okay.
So you can sort of skip even thinking about the progesterone receptor for this question.
And if the question were to say, like, is progesterone required for the repression, that'd be
Correct. Yeah, because it's repressed in both cases and without binding to progesterone receptor. Yeah.
So we have control. We don't look at the drug stuff. We don't look at the no drug, right? Enhancer for anything, or we have to be specific with the task.
It depends a lot on how the question is phrased. Like, what is the comparison you're making, right? So this is just asking you, based on these results, which drugs repress the expression in the presence of progesterone. So I immediately just look at the even lanes, right? So the ones plus progesterone, and I look at if TBP is present there. And I immediately know that TBP is present in the no drug, in the drug 2, and not in drug 1 and drug 3.
Okay, and then so like for drug 1, it's basically so like it's methylated, so it's like not ready to go, right? So it's skin heterochromatin and TBP isn't there, right?
Yes. Yeah, so it's not being expressed. Okay. Okay, so just making sure there's no new words in this question. Okay, based on these results, the addition of progesterone causes which of the following to happen in the no drug control cells? So now we don't even need to look at the whole right side of this, right? We just focus in on the no drug and what happens on the addition of progesterone. So does the progesterone receptor move from the cytoplasm to the nucleus? What are the two types of nuclear hormone receptors? The heterodimers and the homodimers, right? What's the difference between them? Yeah.
The homodimer is in the cytoplasm and the heterodimer is in.
Yes, and the heterodimer is in the nucleus, and it is always bound to the DNA. It's not always on, it's always bound to the DNA, right? So is this a heterodimeric receptor or a homodimeric receptor? Homodimer, right? So in the presence of progesterone, does it move from the cytoplasm to the nucleus? Yes, right? So, and you detect that here by the fact that it is now bound to the target gene. Yeah. If it was a heterodimer, it would be bound in the absence of progesterone also. So the heterodimers are always bound. They are constitutively repressing expression. When the hormone binds, it switches from a repressor to an activator, but it doesn't change binding. It stays bound to the DNA. So it would be bound in both the minus and plus conditions, but it would go from a repressor to an activator. Whereas the homodimeric ones are sitting out in the cytoplasm. They are always activators, but they aren't on until the ligand gets in, the hormone gets in, they dimerize, they go into the nucleus, and they turn on expression. Okay. The progesterone receptor binds a progesterone response element in the BCL2 promoter. So you basically just check whether the gene is now on or off, right? So like you check whether the gene is on or off, which would be acetylation TBP, and you check whether the progesterone receptor is actually bound to the DNA, which it is, right? So it's physically bound to the DNA, right? So that indicates that it is binding a progesterone response element in that promoter. Because it's bound to the DNA, and because that's the way these function, right? So they function by dimerizing, going into the nucleus, and binding all of the progesterone response elements they can find. So this is telling you that there is a progesterone response element in this promoter, right? Because when you do the pull down of the progesterone receptor, you get that DNA.
Let's say if the TBP was present and then it got acylated, it wasn't.
You would still say that it's active. If TBP is binding, it's going to be active. You just, for whatever reason, either your pull down with H3K9 failed in that case for some reason, or that gene has some weird expression regulation that does not rely on H3K9, which is sometimes the case. We give you a very simple picture of histone methylation and acetylation. It is very complicated. Histones are methylated and acetylated at a lot of different places. You might pull down on H3K9 and not get a gene, and it might be one of the most abundant genes in the cell. Yes, so TBP is completely necessary for transcription, and it's sufficient. So if TBP is bound, you can immediately know that that gene is being expressed. Yeah.
So if we didn't have a TBP, like you just said, that would mean that something was wrong and it wouldn't be being expressed, right? So that would like mess up the whole thing.
If TBP wasn't there in the no drug,
Yeah,
then it's not being expressed. Yeah.
Okay, and so then, but like you said, so it's the homodimer is going to be methylated, right?
Yes, and if it was a heterodimer or if it was a homodimer, it would still be methylated without progesterone, always. So the heterodimers and the homodimers do the same thing. They convert the state of the gene from off to on. They just do it through a different pathway, right? The heterodimers are always repressing until they bind the ligand. The homodimers are just out in the cytoplasm until they bind the ligand, and then they come in and they turn on expression, which means in both cases, without progesterone, whether it's bound by a heterodimer or just missing a homodimer, it is going to be methylated.
So if you had another methylated by the drug positive for the no drug, that means it would have been a homodimer, right?
No, so the plus minus is not the drug. The plus minus is progesterone. The drug is either there or not there in both of each two sets, right? So the first two are both no drug, and then it's minus plus progesterone. So if we added progesterone, right, and the progesterone receptor still pulls down in both cases, right, but we're not getting expression with adding progesterone, then there's some other regulation that we don't understand there. It's not functioning like a regular heterodimer in that case. Yeah.
So, which of the proteins are we looking for when we're trying to determine what kind of nuclear hormone receptor it is?
You would look directly at the pull down of the receptor itself and see whether it pulls down in the absence of the hormone.
And if it does, it's heterodimer?
Exactly. Yep. Because the heterodimers are always bound. So even if you don't have the hormone, it's still going to be bound and it's still going to pull down the promoter in the ChIP assay. And then you add the hormone and it stays bound, it just becomes an activator, whereas it was a repressor. Yeah.
So the heterodimer is always going to be, we're always going to have something.
If this was a heterodimer, the whole top lane would be black, right? Every single one of those would have a band.
So the methylated for the no drug and the that is showing us, what does that exactly tell us? Like, that is a homodimer or heterodimer? Like that specific one.
Nothing here, nothing tells you here about it being a homo or a heterodimer better than just looking at the top lane, right? The top lane tells you whether it's bound in the absence of progesterone or not. If it's bound in the absence of progesterone, then it's a heterodimer. If it's bound only in the presence of progesterone, then it's a homodimer.
Okay, the methylated you said is going to be for both always there for the.
So the methylation is going to be there when the promoter is inactive, which in the case of a heterodimer is going to be when the hormone isn't there, and also in the case of a homodimer, it's going to be when the hormone isn't there. When the hormone is there, it will, if everything is functioning normally, no longer be methylated, get acetylated, recruit TBP, and begin transcription.
So in this case, it's a very
Because it's only present in the positive. Yes. If it was present in the negative, it would be a heterodimer. Both of them are going to be present in the positive.
So did we read this like the gel? Like this side or like this side? Like
These are all distinct gels. These are all separate gels.
oh, by lane. Okay.
No, like, see how they're boxed off, right? You have to do an independent experiment for each of these, right? So you pull down the DNA using an antibody against this protein, right? Isolate the DNA out from that, PCR amplify it, and then run it on a gel, right? You can't, like, each of these was from a separate gel. So each of these will go on some, so like, each of these has their own gel, and like one of them looked like this, right? And then experiment 2, it looked like this. Then experiment 3, it looked like this, right? So like, you have many, many gels, one gel for each of these, right? And then for convenience, whoever makes the figure cuts out a box of this, like just in Photoshop, right? And then aligns them all like that. So they're each independent gels, taking a snapshot of just the region of interest, just the correct size, and aligning them like this so that you can read what experiments were done for each. And it's all going to be the same size because you are PCR amplifying a particular region of the primer, right? Or of the promoter, using particular primers that will always give you the same size. If there was something here that did change the size, they would include that, and they would change the way this was presented. Yeah. Whether it's a homodimer or a heterodimer. So once you figure that out, right, heterodimers are always in the nucleus, they're always bound. Homodimers are out in the cytoplasm until the hormone is present, in which case they dimerize and go into the nucleus. And this is specifically for nuclear hormone receptors, right? So like, in this class, that's always how the nuclear hormone receptors are going to function. Okay. All right. Okay. Which of the following are consistent with these ChIP results? Is drug 3 blocking the ligand binding domain of the progesterone receptor? Yes, right? So we're looking at this, right? What's ordinarily supposed to happen is you add progesterone, it binds to the receptor dimerizes, goes into the nucleus, and now it's bound to DNA. You can do your pull down, you can get your signal, right? So this is a consistent way that drug 3 is stopping this because, right, now when you add progesterone, you don't get it binding, right? So that is consistent. You blocked the ligand binding domain of the progesterone receptor. This is the readout you would expect to see. Okay. Drug 2 results in progesterone-independent regulation of the BCL2 gene. Yes, right? So with and without progesterone, we are getting expression in drug 2. So drug 2 has broken this system and made the BCL2 gene constitutively active. Drug 1 blocks co-repressors from binding the progesterone receptor. Is that consistent with this? So what would it mean if drug 1 blocked co-repressors? Would you get more expression or less? More, right? So co-repressors are going to repress. If drug 1 blocked their ability to get there, now it would be more active.
But how can it self?
It's in the drug 1 case, it's not active, right? So if drug 1 blocked co-repressors, you would see acetylation, you would see TBP, right? Okay. So if drug 2 blocks the ligand binding domain of the progesterone receptor, this is essentially asking the same thing as drug 3, right? If that was true, it would look like drug 3, where adding progesterone does not lead to it going into the nucleus. Clearly, adding progesterone, it still is able to dimerize, it still is able to go into the nucleus, so D is not consistent with these results. Any questions on this one? So for D, right, it's asking the same thing as A, but for drug 2, right? So since it's still able to bind to the promoter in the presence of progesterone, it's clearly responding to progesterone, so there's no way the drug is blocking the ligand binding domain. Okay. To identify all the genes associated with TBP in uterine cells treated with drug 2, you perform a ChIP experiment using antibodies for TBP and next-gen sequencing. Which of the following would you observe? Yeah.
Wouldn't it be A and then B and B, right?
Okay, so A and B are good, right? So higher levels of sequencing reads for genes associated with TBP, right? That's literally what you're measuring with this, right? You're pulling down on TBP with an antibody. You're pulling all the DNA that it's bound to with it, right? So you will get sequencing reads for genes associated with TBP. Sequencing reads that map to many locations in the genome. Where is TBP binding in the genome? Everywhere that's active, right? So everywhere that is having active transcription will be bound by TBP. So there are going to be tons of locations in the genome where TBP is binding. Do you detect alternative splicing by a ChIP-Seq experiment?
Wait, no.
Why not? Yeah.
Because with the chip, we're just doing the histone breaking up, right, and seeing if that's expressed.
So we're starting with chromatin, right? We're getting chromatin. We're doing a chromatin immunoprecipitation. The chromatin is not the RNA, right? The only way you see alternative splicing is if you do an RNA-Seq experiment. You won't see alternative splicing from the DNA because the DNA doesn't get spliced. Okay, and then C, lower levels of sequencing reads for genes associated with TBP. No, that's counter to what the experiment is doing, right? The experiment is looking at what's bound to TBP. You would see higher reads for the TBP-bound proteins, or TBP-bound genes. Any questions on this? All right, last one. If you use CRISPR to change the template sequence in the gene for telomerase RNA from that to that in lung tumor cells, which of the following would result in this change? So how does telomerase work? What type of enzyme is telomerase? Yeah. So telomerase extends telomeres, right? So telomerase is a reverse transcriptase that takes an RNA template and extends the telomere DNA from the end of chromosomes. Yeah, it's very iffy, right? So like the ASEs. A lot of the ASEs are hydrolysis reactions. Technically, any enzyme, the scientists are free to just end it with A's for fun. But historically, right? Originally, it was for hydrolysis reactions. But yeah, so A's really just means enzyme. Okay, so telomerase is a reverse transcriptase, right? It's going to use this RNA template to extend those, right? If you mutated the RNA sequence, right, what would then happen? Would telomerase base pair to the telomere? Not anymore, right? So the base pairing is going to be disrupted. Would the telomeres be maintained? No, right? So because it can't base pair, it can't extend those telomeres. Would the tumor cells stop dividing? Yes, right? So that telomere extension is necessary because you lose the ends of your chromosomes a little bit every time you divide, right? The reason tumor cells upregulate telomerase is so that they can protect their chromosomes on subsequent rounds of division. Would the telomere sequence change to match the RNA? No, right? That's way too difficult of a specific mutation for the cancer cells to do. It would be really impressive if they could do that, but they can't, right? So that would have to be multiple specific base changes in that one. Okay, so this is A, B, and C. Any follow-up questions on this one? All right, thank you guys. Yeah, feel free to come up and ask me individual questions if you have anything specific, and I will see you Thursday. Hello.
I have a question, last question.
Uh-huh.
You said like you would be really impressive with targets, so that means the mutation is